And we're going to go ahead and get started. Thanks so much for being here with us. Whether you're online or in the new auditorium, thank you so much for being part of our research seminar here at Hatfield. My name is Cinnamon Moffett. I'm the research program manager here at Hatfield. It's part of Oregon State University, located in Newport, Oregon. I will be your host for today. A couple of announcements. If you are online, we have your screen shares mics turned off, but we're hoping that you'll ask us any questions by using the chat function. We have somebody in the room, Michael Banks, who is going to help us moderate those chat. You are also welcome to raise your digital hand and we can unmute you at the end and you can ask your questions that way. For those folks that are in the room, if you have questions, again, we'll get to them at the end, but the main thing is to include our digital audience. You also need to get to a mic to ask your questions. You have two options. You can either get to the mic stand that's right here, or you can raise your hand and I'll bring you the mic that I have. So either way, but that way the folks online can hear the questions that are being asked. A couple of little announcements I wanted to make. If you don't know, Marine Science Day is Saturday. So, and it's supposed to be not a day like today. It's supposed to be rainy. So if you'd like to join us, we are online this year again, but we'll have virtual live events streaming all day between 10 and 2. One of them being a shark dissection by some folks that are in this room. So check that out if you'd like. There is a flyer up here if you're unsure about that event. The other promo I wanted to do is that Hatfield has a team doing Out of the Darkness suicide prevention and awareness walk on Saturday as well in Corvallis. So if you're interested in that, you would talk to Melissa. You either can donate or you can walk. So if you want to be a part of that, we have a busy Saturday. I also wanted to promo next week's seminar. We have Kyle Newton, who's in the room, who is part of the Big Fish Lab and is a new member of Hatfield, and is going to be talking about cryptic senses of fish using wildlife models, species, and machine learning to address super rad ecological questions. So if that doesn't entice you, yeah, it'll be a good talk. So I hope you can be there. Well, last little promo, the green team here at Hatfield is also working with Solve the day after Earth Day. So that is April 23rd. So if you're interested in doing a river cleanup along the Yaquina River, come talk to me after or check out the flyer or email me. We can get you hooked up with the green team and you can help do a river cleanup. But with that and all of that, we're also recording today's event for folks that are online. If you want to share it, you're able to go to the past seminar page and you'll get a recording. For those folks in the room, past seminar page on our website, you can get that recording and we can share that out. But I want to introduce today's speaker, and this individual was invited today by our very own Taylor Chappell as part of the Big Fish Lab. So Taylor, I'm going to hand this to you.
Thank you, Cinnamon. And I'll just clarify about the dissection at Marine Science Day. It's actually going to be 2 sharks. So we just doubled the fun. But today it's my pleasure to welcome Dave Cade. Dave and I have known each other for quite a while, back when I was at Stanford and Dave was there doing his PhD. I was trying really hard earlier to remember all the years of all of his degrees, but I'll basically give a very quick background is that Dave actually has his bachelor's in mathematics and then got a master's in education, and was a teacher for a long time, and then went back and got his master's in science here at OSU. So he is an alumnus of OSU, and then did his PhD at Stanford, a postdoc at UCSC, and then back to Stanford again for another postdoc. So he's definitely gone through a number of very cool different experiences. And he's going to talk about some of that awesome research today. One of the things that I think most fascinating and coolest about Dave's research is that the work that he does really transcends the species. And so I'm sure he's going to talk about marine mammals today, but a lot of the cool technology and analyses and things that Dave does with the marine mammals, he also does with other species. And just today I was working on a package in computer language and went to Dave's wiki site where he's done incredible amounts of work to bring the really complex math and computer coding to the rest of us. To make these types of analysis and studies possible. So with that, I'll let Dave take it away.
Thanks, Taylor. That's a great introduction. I appreciate that. Let's see if I can get rid of this upper bar here. I don't know if we can do that or not. Hide video panel. My mic, I think, is on. Everyone can hear me? The high floating meeting controls. Not here. 3 dots. Hide. Yeah, nice. Thank you. Right on. All those degrees don't help you. Thanks. And the other thing that Taylor didn't mention is that my first experience with marine mammals was actually with the Marine Mammal Institute. It was actually in 2007 while I was a teacher. I came up and took Jim Summich's marine mammal course. And I have a great video, if anyone ever needs it, of Jim like blowing up like a sea lion lung, the stellar sea lion lung, just like freshly dissected, and just have his lips all over it. It was great. So that was the inspiration for going down this path. Thanks for having me here. Thanks for inviting me. I'm going to talk about a number of things, and a lot of it will be, we're kind of bouncing back and forth. I'm going to do some new things that I haven't talked about before, so bear with me if there's too much data or too many graphs, and you can tell me that later and give me some feedback on that. But like Taylor said, I am currently at, let's click back over to here. There we go. Jeremy Goldbogen's lab, did my PhD with Jeremy Goldbogen, and then I was now doing a postdoc with him. So I'm going to flip back and forth a little bit between some of his work, kind of setting up the work that I do, and then some of the work that I'm doing as well, and kind of leading to this idea of size limitations on engulfment filtration feeding, and kind of more generally, just what are some of the size limitations on bulk exploitation of patchy prey, which is where my intersection here this week kind of comes from. I've been here, as Taylor said, working with the Big Fish Lab, working with Taylor and Alex. We're starting a basking shark collaboration. We're going to go out to Ireland and do some work on this large filter feeder that is kind of this cross between a shark and a whale that we're really excited to work on. So this will come into the talk a little bit later on, but mostly I'm going to be talking about rorqual whales and baleen whales, and specifically minke whales a lot, which is what this guy is right here. I want to just to talk about what I'm going to talk about. To outline it, we're going to talk about the difference between particulate versus filter feeding. We're going to get into specifically in cetaceans. We're going to get into some of the unique aspects of rorqual whale engulfment filtration feeding. Really, what is it that makes these animals so unique? We're going to talk about the evolution of this feeding mode. We're going to talk about how feeding rate and prey density kind of interlock to really allow this feeding modality. And then what we're trying to lead to, the new part here, is this idea of how giganticism and bulk filter feeding are enabled and kind of co-evolved over time. So we're kind of going to lead down to that idea throughout the course of this talk. Before I really get started, I want to give a big thank you to a lot of the people I work with. I can't do any of this work by myself, really. Like, you can't tag a whale by yourself. And so just thank you to all the organizations that have supported us and have worked with over time, and all these field teams that are really kind of a critical part of the work that I do. So I'll do some individual shout outs as we go, but just want to give a general thank you to a lot of the different groups that we work with. Okay, so let's start with this idea of particulate versus filter feeding. This is a killer whale in Norway. This is a paper that I think just got published this week in Marine Mammal Science. And killer whales are kind of the penultimate, sorry, Taylor, the penultimate particulate feeder in the ocean. They really are like these massive hunters that are kind of the apex predator in the ocean. But these are killer whales feeding on herring. And so they have to eat a lot of herring in the ocean to really account for their giant size and make up for their metabolism. And they do that in a number of really interesting ways. By, as you see here in this video, working together. They work together and they make these really complex loops to help herd and corral prey into these balls that they can take advantage of. And even then, even when they corral all this prey, they still have specialized feeding techniques. This is right there. If you can see that, I'll even back it up again. This is a tail slap that animal does to actually stun all those herring so that they can come and pick them off after they're already dead. So there's really a lot of strategies they have to use to feed on these bulk, these animals that all school together but are so small, you have to kind of do a lot of work to get there. These are common dolphins in Southern California, and they are doing a similar kind of work. They're working together. They're doing this cooperative herding to make this nice ball of anchovies, in this case, that they can feed on. You can see that ball of anchovies there, and we'll come back to that anchovy school. But in most systems, these big predators, right, have to use fast speeds to overcome the performance capabilities of their prey. But But if you can't be faster than your prey, you often use other kinds of strategies like cooperative feeding and group hunting, like we just saw with the killer whales, or weapons, like we just saw with that tail slap. So some other strategy to take down prey that are more maneuverable than yourself. So that brings us to this idea of filter feeding. This is a barnacle on a whale. It's a filter feeder on a filter feeder. And filter feeding is highly efficient. And it's really highly efficient because you have a lot of energy in for very little energy out. Like this barnacle isn't doing anything, but it's getting a lot of energy as it gets movement through the ocean to just kind of sit around and watch what's happening and to collect the prey that's in the water column. Similarly, sessile invertebrates have a similar idea. They can just sit around and be filter feeders. And filter feeders, though, also can be mobile. This is a krill filter feeding, which is the prey of a lot of these animals. And then once you start actually measuring some of the biomechanics of these filter feeders, there's some similarities no matter what your size are. So anchovies actually slow down considerably when they're feeding because it takes more energy to open their mouth and filter feed in there. So even though you can just open your mouth and food is swimming through your mouth, there's some energetic costs associated with that. So that's similar to what right whales do. Right whales have to slow down when they're filter feeding. They have that continuous ram filtration style where they'll dive down to the depth of copepods and then slowly filter through that water column. And whale sharks do a similar thing. They decrease their tail beat frequency when they're filter feeding because they're slowing down. They have to have more drag as they open their mouths to feed. So what these animals all have in common is that they're slowing down to feed or not moving at all in some cases, right? So they're moving really slow. Contrast that with our rorqual whales. So let's go back to our fish school here. This is the common dolphins herding the fish schools in Southern California. There's our fish school. Unfortunately for them, this picture was taken off the back of a humpback whale, which is coming to engulf this big fish school all at once. And you can see it's a really aggressive maneuver, right? It's a really fast-paced maneuver. Here's another example. This is Monterey Bay, also a humpback whale feeding on anchovies. All these sea lions are trying to pick off individual anchovies, one by one by one by one. But this humpback's going to come in there really aggressive, about 4.5 meters per second as it kind of thunders up through the edge of that school and gets one of the bites off the edge there. So it's a really energetically expensive process. It's different in the filter feeding realm to have this really energetically expensive process. So just to kind of clarify what we're talking about here. So there's the continuous ram. Baleen whales can have three clades, right? There's the bowhead and right whales, which are the continuous ram filtration feeders. Maybe what your first thoughts of a whale were when you were a kid, just kind of slowly grazing through the ocean. There's the gray whales, who are the bottom feeders, primarily suction feeders along the bottom, and they can feed in the sediment by actually pulling up invertebrates. They can do some water column feeding, as the Gem Lab will tell you, but they're primarily bottom feeding. Versus the rorqual whales, which we're going to talk about primarily today. The rorqual whales are this engulfment filtration feeder. So this is a fin whale in California, and it has this lunge feeding has these three components of acceleration up to speed, about 4 meters per second, engulfment up to your body mass in water, and then filtering. And filtering can take the longest component of all of this. For a big fin whale like this, the filtration period can be 40 seconds to a minute long or so. So there's these three components, but the longest component is filtering. And we'll come back to that idea just to kind of keep that in mind, because that's going to change when we get to minke whales. So let's start getting into that idea of what makes rorqual whale filter feeding so unique and so exciting. And I know a lot of you guys probably study this and think about these ideas, but I want to kind of review it for those of us who don't. So this is a blue whale feeding on krill in California. And the thing I want you to, the most impressive piece about this, of course, is that giant tadpole-like shape where it's engulfing up to its body mass in water and krill that then it filters out. It's that engulfment filtration feeding process. And that pouch can be really, really big. This is a dead whale, and the gases in this whale have expanded that ventral pouch to really kind of put pressure on that. And it's extended up to 4 times its resting length, those muscles and blubber in there that's extended out there. But that's not its natural state. It's not just a passive process. When those whales are dead, if they don't have those gases there, like in this whaling example, that tissue is relaxed. And when it's relaxed, it's just kind of like this accordion tissue that doesn't have tension on it. It doesn't have those muscles kind of actively contracting it. These muscle tissues are amazing. It's a really unique adaptation to be able to have this pouch here. It's there's 2 layers of muscle tissue below the blubber there. And the oblique stratum there, that OS, has this really cool cross pattern where the muscles are both at a 90-degree angle to each other and crimped, so that when that whale expands its VGB, when it expands that buccal cavity, it has that ability to then contract those muscles again. And every time that they're doing this, their muscles can contract up to expand 1.5 to 2 times their resting length. And if you can imagine like your arm doing that, right? Like expanding to twice its resting length and still having your muscles work. So it's a really unique set of musculatures that they've had to evolve. One of the things we're going to talk about as we go through today is the evolution of these. And one of the things that we don't have a fossil record of is when this accordion-like VGB ventral groove blubber has started to appear in the fossil record. So because it's hard to fossilize. So that's one of our missing gaps here. Okay, one of the other things you might notice about rorqual whales is this big curved jaw there, that lower mandible. And what's really unique about those jaws is that they're hinged. So right now in that minke whale there, they're in the rotated up position, but as they feed, they can rotate their lower jaws, their lower mandibles out and make a really big net, essentially. Here's a humpback whale coming up, and you can just see the size of its mandibles rotated all the way out as it's engulfing as much as it can. And that allows it to have that really big pouch and really engulf that 100% of its body mass. So have these specialized jaw joints in there to allow for that. They have this unfused mandibular synthesis up in their chin that allows that rotation, as well as the hinged jaw joints in the back. And one of the things that controls all that is this specialized organ that Nick Pyenson and Jeremy Goldbogen and Bob Shadwick dissected and found this specialized organ right in the chin of these, right in that unfused mandibular synthesis that seems to be a sensory organ that helps to control this whole process. So it's another really unique adaptation that kind of enables this lunge feeding process. And the final thing to talk about is this idea of filter feeding in general. So after they do this big engulfment, they have this big pouch, this big mouthful of water. They've controlled that whole process. They still have to get rid of that water. And that's where it's filtering out the lip there. You can see that exposed lip with the baleen kind of hanging down from the upper jaw there. And the water all has to get pressed out of there. And in this blue whale, that can take about a minute or so to force out that 100 tons of water. And one of the things we don't really have a full grasp on is exactly how that filtration process works. People used to think in the old marine mammal literature, a lot of the time there's this idea that, oh, the water is forced out, the water's engulfed, and then it's forced out through compression, and then the tongue will like clean up all that krill off the baleen. But if you think about that, that's a recipe for getting things trapped in there. It's like your colander, right? Like your stuff's going to get trapped in that colander and you have to clean it out. And that's not a really efficient system. So what we actually think is happening more likely is more of a cross-fill filtration system where the water kind of gets sloshed back and forth and the prey ends up aggregating towards the back of the mouth with the water going out the sides. And we think that's what's happening, but it's hard to study that for sure. Okay, so let's talk a little bit about how this feeding mode developed. So, oh, nice slow animation. So filter feeders have been around in the ocean for a long time. There's this 100 million year dynasty of giant planktivorous bony fishes in the Mesozoic seas. So a really long history of big bony fishes filter feeding, what we think are filter feedings. They have big jaws that are morphologically similar to some of the big filter feeders we have today. And then pretty soon after these animals went extinct, there's this transition to the big filter feeders we have of today, the manta rays and the basking sharks and the whale sharks, and critically the whales, the filter feeding whales. And whales first evolved from land-based ancestors, dog-sized ancestors, all carnivorous ancestors of artiodactyls in the Eocene, about 40 to 50 million years ago. But they weren't filter feeders then. They're these predators that are kind of in these shallow seas. The clade of mysticetes, the baleen whales, started to evolve about 35 million years ago or so, but they still weren't filter feeders originally. The first mysticete ancestors, which we know of from certain bones in the skull, but did not have that characteristic baleen, were about 35 or 40 million years ago. And then the first baleen didn't appear in the fossil record or evidence for baleen based on jaw morphology and holes in the jaw until about 23 million years ago or so. And it's interlocked with this idea of giganticism. So you have this kind of these two interlocking plays here happening with whale evolution, with animals getting big and with filter feeding and the type of filter feeding they're doing. So let's look at this idea of giganticism. I mentioned that all of these animals are big. You have big whale sharks, big manta rays, big bony fish, but all of these animals are in this 2 to 9 meter range, which is big. That's a big animal. A 9 meter animal is from here to that staircase over there. That's a big animal. 2 meters is also pretty big. If it's bigger than me, it's big. But that range, right, 2 to 9 meters, that's about the size of the smallest rorqual whales, the smallest filter feeders we have right now. So something happened that allowed the really big filter feeders to develop. This is a minke whale in the Antarctic. This data, all the whale lengths I can show you guys are from all done by Casey Berlick in the GEM lab, postdoc when he was, most of it was his work when he was a PhD student at Duke. So yeah, this is a funny picture of Casey. But so all the lengths you see throughout are all his work where he did these direct measurements from photogrammetry. This is me in the boat here with my antenna to find the whale, which I don't necessarily need right now, but because I found it. So minke whales are small. They're the smallest baleen whale, but they're about the size of all these historic ancestors. And so how did mysticete evolve? What's the history here? Well, this is a diagram now from a Slater article about the size of animals from the fossil record based in time. So we're blowing up that last 30 million years there. You can see that first baleen on there. You can see where the first mysticetes, the crown mysticetes started to appear. And you really didn't get like giant whales until bigger than 10 meters, bigger than that historical record, until about 10 million years ago or so, 8 to 10 million years ago. You have some of those ancestors that are about 10 million years old. And you see this kind of general trend of getting bigger and bigger and bigger. And like I said, we don't know all of the stages here. We don't know exactly when VGB evolved. We don't know exactly when that specialized chin mandibular synthesis evolved. But the other critical thing to note is that even when we say big animals bigger than 10 meters, nothing really bigger than 12 or 15 or 22 meter giants that we have now until really recently, really only in the contemporary oceans where we have these really, really big animals. So we think most of those animals evolved in the last 5 million years or so, pretty recently in the fossil record. This is a small blue whale, a 22 meter blue whale. And here's that minke whale for comparison, just to kind of get in your head the kind of sizes that we're talking about when we're talking about these animals. And blue whales famously can get up to about 30 meters in length. Commonly today, you'll see 24 to 26 meter whales. Okay, with us so far? Doing okay? Can you hear me in the back? All right. All right, so let's talk a little bit about the type of filter feeding. The other thing to remember about all this history is that most of these animals were ram filtration feeders, that they were doing that slow forward swimming, opening their mouths, swimming through the water column, taking whatever happens to be down there. So basking sharks are in that group of animals that we're curious about because they live in cold water and they somehow get enough food by swimming slowly with their mouth open here to survive, these big animals. And we compare that to our lunge feeding. So how does lunge feeding fit into this story? How do we get these really big lunge feeding animals? It gets to this question from a recent whale shark book. This is from last year. Devin Pierce asked this basic question, right? Are animals able to become planktivores because they are so large, or do large sizes result from planktivory? It's chicken and egg here, right? What's coming first? How are these animals all fitting in together? So let's get into this idea of prey density, how prey density and feeding rate kind of interlock here. So these are blue whales off the coast of Monterey Bay. This was in 2017 when we were seeing aggregations of blue whales up to about 50 in an area of about a square kilometer. So a lot of animals in one area. But historically, it's tough to study marine mammals, as you all know. They once they go in the water column, once they disappear beneath the waves, they're kind of gone to human observation. They're difficult to study once they disappear there, and the surface is only such a small portion of their lives. So in the 20th century, the tools that we could use were restricted to things like anatomical studies from dead animals that were either killed as part of the whaling industry, or washed up on shore. You could do some stomach content analysis. You could look at surface observations and surveys, and people really have been fascinated with whales for thousands of years, and these are kind of some of the best tools that we have. Starting in the 21st century, we have some really ramping up some new technologies here. We have the chemical signatures. We can do DNA analysis and stable isotope analysis kind of on the regular. So these really cool technological tools. We have enhanced surface observations, like some of those drone videos that I've been showing you guys, or that you guys are very familiar with. And then we have the in vivo underwater observations. And that's where I do a lot of my work, which is the kind of the biologging technique, where we can study the predators. And then we have like fisheries acoustics, which is what I studied here at OSU, where we can study prey. So combining those two ideas to look at in life, what's happening under the water surface, this is kind of where I do most of my work and what most of the tools I'm gonna talk about for the rest of this trip, the rest of this talk. So this is a fin whale tagging a fin whale. This idea of biologging is the use of miniaturized animal attached tags for relaying data about an animal's movement, behavior, physiology, or the environment. And these tag technologies have come a long way since the first kitchen timers, essentially, that they put on seals. Oops, skip the good part. So this is, again, a blue whale in Monterey Bay. The way we attach these tags, this is a small boat working with Cascadia Research Collective, one of our good collaborators. That is me on the front there. We have a long pole, about a 6-meter pole with a suction cup attached tag on the end. And basically what we do is wait for these animals to surface. With whales, they have to breathe. And so we wait for them to breathe. And we have a quick approach with a nice attachment there, so that right there at the end of that pole is this suction cup attached tag, and that tag will stay on for 6 to 24 hours or so, and then pop off and float to the surface, and then we'll recover that data. It's logging all that information on the tag itself, and then we have to recover that package to getting the information from it. And then what's nice about that is, so then once we do that one approach, we are done with that whale. We let the tag do the work after that. We let the tag kind of sit there and log that information from those animals, and we'll come back and recover it later. So what is it logging? Well, what that tag is logging is that fine scale motion of that animal underwater, that orientation, motion, and position of that animal underwater. So this is a depth profile of this blue whale there, going down, doing a number of wiggles here, which we'll talk more about. That's our signature, one of our signatures of feeding. And then we can also log not just depth, but what is that animal actually doing and how is it actually moving? We have its orientation, its pitch, and its roll, and its heading, all from these same kind of sensors that you might have in a smartphone that allow you to basically to know which way your phone is facing, whether it's horizontal or vertical. It's using a gravity sensor and a compass to tell the orientation of that animal underwater. So a lot of the work that I do that Taylor talked about is to develop the tools to understand the information that comes off of these tags. The tags that we primarily use are these video tags. So we'll often either have 1 or 2 cameras on these tags. And then there's the raw sensors there, the accelerometers, the magnetometers. We have GPS, which of course only works at the surface. So you have to have tools for integrating between surface hits. We'll have hydrophones and then video that can record up to 9 hours of video. So this is a video from a blue whale. And we're going to start looking at some of the differences between how blue whales and how minke whales feed. I want to orient you a little bit. This is a blue whale, but this is a tag that's going to slide down. This is its right pectoral fin on frame right there. You have on the data down here, you have the blue line is its depth and the green line is its speed. So we're right, and that red line is where we are in the video. So we're right at that high speed event right there. And because it's high speed, and this was an early version of a tag that was prone to slipping, the suction cups were not quite as good as we have now. You'll see this tag kind of slide down the belly of this animal as it starts to feed, as it goes to this high acceleration feeding event. And then over here, we're going to show a minke whale as a comparison. So you can kind of see both of these animals feed. And I show these 2 videos because we're looking at the underside of both of these animals. And you can see that big ventral pouch expand. And the thing to notice here is that this whole time that this blue whale is going to be filtering for about a minute or so, this minke whale is going to be lunging and lunging and lunging. It's about a 5-meter minke whale. So that means it's engulfing about 3 tons of water that it then filters in about 5 seconds. It's like almost a ton of water per second for some of these animals. And it just keeps doing it and doing it, doing it. And meanwhile, the blue whale over here is just kind of doing that one filtration. So the advantage of being small is that you can feed more and more and more often. You have these high feeding rates. The advantage of being big is that you can engulf more at once and you don't have to feed as often. Okay, so how do we study that interaction of predators and prey? So we have this blue whale, it's diving down, it's doing these loop-de-loops, and we can identify those feeding events, those foraging events. We can use fisheries acoustics to look at how these whales and how they're interacting with prey at different densities and distributions. And we can look at how they're interacting with fish schools using some of these same technologies. And it allows us to start studying this idea of foraging efficiency. So this idea of how much energy are they getting in with these giant gulps, and how much energy are they putting out with their with these loop-de-loops? How much do they have to spend? How much energy do they have to spend at speed to engulf all this prey? So throughout the rest of this, we're going to use this symbol lambda to represent energy in. It's basically your intake rate. How much food are you getting per unit time? So we'll just kind of keep that in your mind as we go through this next step here. So lambda, though, that energy in, you can break down that intake rate into its products, right? The amount of energy you get is going to be related to the prey density, the patch density and distribution, and the foraging rate. How many times are you lunging per unit time, times how much food are you getting with each gulp? You have the baleen whales, these rorqual whales are this kind of unique modality where they are kind of like a raptor, right? They have this raptorial filter feeding style where they engulf a huge amount of water. It's a really chunk of water, but then they do it again. So it's a combination of raptorial feeding and filter feeding that makes them a unique case study. And you can break down this foraging rate into its components as well. What are the thinking about now for the rest of this talk about the limitations on this foraging rate? There's biomechanical constraints. How quickly can that minke whale lunge again and again and again? There's physiological constraints, and then there's environmental constraints. So the biomechanical constraints are lunging. How long does it take for that animal to do a lunge? And how much water can it engulf on any given time? That's a biomechanical constraint. That's each of those individual lunges. The physiological constraint is the surface recovery and transit time. All of these marine mammals are diving to depth. They have to hold their breath while they're doing dives over and over and over again. So how much time they have to spend at the surface recovering is a big constraint. And the bigger you are, the less relative time you have to spend at the surface. So that's an advantage to being bigger. And on the minke whale side, that's a constraint. You spend less time at depth overall. And then there's this idea of environmental constraint. That's how much time it takes you to find one lunge to the next. Part of that interlunge interval, how much time from lunge to lunge to lunge is maneuvering yourself to find the next prey patch. And if you're in a really big prey patch where there's just food everywhere, I can show you later some idea of these giant prey patches where animals are just lunging every which way and there's no gap between their lunge feeding events, versus some of these animals have to actually maneuver and find the next prey item. So that environmental constraint of how much time it takes to find your next meal. This is a giant prey patch. So you have this giant prey patch here where that animal can just kind of lunge again and again and again without really thinking about where that next patch is going to be. So this leads to some work where we compared, because I'll step back, you can do this analysis at a lot of different time scales. One of the time scales that we look at a lot is this idea of an individual dive. How much food are you getting on an individual dive? So this top plot here is a blue whale dive profile and corresponding to its prey density. The bottom plot there is a sperm whale, and you can look at these individual dives for a blue whale versus an individual dive for a sperm whale and look at its energy in versus its energy out for each individual dives. Look at its efficiency of these both apex biggest animals in their clad, the blue whale and the sperm whale, and look at what is their energetic efficiency in each of these situations. So working with Jeremy Goldbogen and Danuta and a lot of others, we put this all together into this idea of foraging efficiency of odontocetes and mysticetes. If you look at odontocetes, the toothed whales, one of the things that we noticed, if you look at their prey and look at their efficiency on each of these individual dives, is that as animals get bigger, actually decreasing their overall efficiency. And what that points to is a limitation in body size for these animals. So why is there not an animal bigger than a sperm whale? Well, they're not getting as much prey as they need to be energetically efficient. There's some constraint there from an energetically efficient standpoint for these odontocetes. So there seems to be this kind of overall size limitation there. Now let's look at those baleen whales, specifically the rorqual whales here, these lunge feeders. What's their efficiency look like? Well, if you graph these animals, it has the opposite trend. These rorqual whales are increasing on a dive-by-dive basis in their efficiency. And not only are they increasing, they're also really, really high. Specifically, there's this big question now. We're getting to our title of our talk here. Well, this does not seem to be limiting the size of blue whales, right? Like it seems to suggest that the bigger you are, the more and more efficient you are. It also asks this question now of why is this the smallest filter feeder? If this foraging modality, lunge filter feeding, is so efficient, more efficient than almost any of the odontocetes on average, why aren't there smaller baleen whales kind of filling that smaller niche? Why aren't there anything in the 2-meter range or the 3-meter range or the 4-meter range filter feeding and engulfing krill in the same way? So that's the motivation for the rest of this talk, which I'm gonna tell you guys about here now. Just checking my time. And so basically this question of why are minke whales the smallest lunge feeding whale? We're going to get to this last question here. We've got about 5 or 7 more minutes here. So we started introducing this idea of temporal constraints, this idea of how much time these different aspects take. And some constraints scale with body size. For instance, gestation, right? A human has about a 9-month gestation period. A mouse, very small mouse, has about a 3-week gestation period between before it gives birth. Big elephant, a 7-ton elephant, which is about the size of a minke whale, has a 22-month gestation period, some of the longest in nature. Killer whales kind of right on that spectrum, right? These odontocetes, these particulate feeders, right on that spectrum. A 3-ton killer whale has about a 17-month gestation period. So how does that fit in with rorqual whales? We have a minke whale, a humpback whale, and a blue whale. Minke whale is about 8 tons. Humpback whale is about 23 tons. Blue whale is about 80 tons. And their gestation periods are about 11 months. Pretty fast for an 8-ton animal. A humpback whale, also about 11 months. Blue whales also about 11 months. So there's some other constraints here that are kind of universal. So even though metabolically you'd expect something like a blue whale to have a really, really long gestation period, it's limited by some of the same constraints that all animals experience, meaning these guys are capital breeders. They have to bulk up and feed during their peak of summertime and then give birth on the seasonal cycle. So they bulk up all that energy and then they give birth, and they bulk up all that energy and they give birth. And that annual cycle is the same as it is for you and I. Same way that animals that only feed at night have to deal with that 12 hours of daylight the same way, whether they're big or whether they're small. They have that same constraint. Another famous example is cellular size, right? Elephants and mice have the same basic capillary size, and so that can constrain your metabolic delivery system, and that leads to some of those metabolic scaling aspects that people love to study. So they have this idea of these temporal constraints that don't really scale. So what's the question then is, what's the right foraging temporal scale? We saw on that dive by dive scale, there wasn't really a limitation to feeding and to filter feeding on either end of the size spectrum. So one of the ways to think about it is not necessarily on a dive by dive scale, but on this thinking about the scale at which an animal is actively choosing to feed. What is their feeding rate while they're actively feeding? And so here's that blue whale dive profile. And instead of thinking about one individual dive, you might think about this whole aspect as a feeding bout, because even though it stops and recovers oxygen here, it's really kind of continuously feeding throughout this time period. It goes up to the surface, breathes, and go right back down to that patch to feed again. So you start to look at like, what is the actual feeding rate of this whole period, which then takes into account some of the physiological constraints and some of these other constraints in its foraging. One of the ways in which these 3 species, minke whales, humpback whales, and blue whales converge is that they have essentially the same time period between dives, between foraging dives. If we plot how much time does it take between one foraging dive and the next to recover at the surface, there's some skew in all these data points, but the critical thing is that right here at about 5.5 minutes is where you have that break where most of the data is to the left of that line, and to the right of that line are kind of these extreme examples. And so you can use this as a cutoff essentially to say like, okay, well, if it's more than about 6 minutes, that animal's probably doing something else. It's either moving to a different location, it's doing something else. On less than 6 minutes, it's probably in the act of still foraging. So we just kind of looked at a lot of data to identify this time period of what is a foraging bout. When is an animal actually choosing to stay in this actively foraging modality? And so that leads to this idea of this whole section being one foraging bout and looking at the feeding rate within that foraging bout. And I kind of hinted at this already, but minke whales feed a lot. They feed a lot. So this is a minke whale during the day. Here's a foraging bout. It's lunging at about 73 lunges per hour. That's almost more than one a minute. It just keeps lunging and lunging and lunging and lunging and lunging. Here's a close-up of that. And this is even regardless of spending about as much time at depth as they do at the surface. So they have about a 5-minute recovery period at the depth, or maybe, what is that, 3-minute recovery at the depth, and then about a 3 or 4-minute dive at the bottom. So they have all these other constraints in their time, but they still have this really high feeding rate. But where it gets interesting is then if you look at minke whale behavior at night. And if you remember that at night, most of the krill, both in this ocean, in the Antarctic, has this DL vertical migration pattern, where during the day, krill is down at depth. And at night, it's coming up to the surface. And what that allows is these minke whales to have really, really high foraging rates at night. So up to about 240 lunges per hour. Every 15 seconds on average, this animal just keeps feeding and feeding and feeding. And partly that's facilitated by this krill at the surface where the animal doesn't have to dive very deeply. It only is diving to about 10 meters there and just keeps lunging and lunging and lunging and lunging and lunging. So let's look at the scaling of that idea a little bit. We have this temporal constraint of feeding. So this is a plot of minke whale body length. We're going to look at all the different body lengths down here. We have lunge rate on the y-axis here. So x-axis is length, body length. Y-axis is feeding rate. So we have those kind of average feeding rate of those animals up to, so that's 28, I think, data points there for minke whales that we tagged in the Antarctic. If we throw blue whales on there, we expect blue whales to be a lower feeding rate, and indeed they are. So we have these 19 to 25 meter blue whales and have these feeding rates more normal of about 20 lunges per hour or so when they're actively feeding. And to round it out, humpback whales are right in the middle. They have this nice, so you get this nice line on a log-log plot that kind of connects these dots, and you have this nice expected range. So you have this increase in feeding rate as you get smaller, decrease as you get bigger. But what were we expecting? What's the slope of that line? And how were we expecting that slope to change based on what we know about rorqual whale foraging habitats? So we come back to this idea where our intake rate is a product of prey density and feeding rate and the engulfment volume. How much food can you engulf with each individual lunge? For the purposes of the rest of this, we're going to actually, even though I love talking about prey density, we're going to assume that on average, the prey density is about the same. Like if there's some environmental limitation of prey density, that's a grant we can write and we can work on that. But we're going to assume that's the same for now. And critically, intake rate is thought to scale generally in the same way that most metabolic rates scale, about with mass to about the 0.75, or we can quibble about what that exponent is. But critically, it's in that range of 0.7 to 0.75, and larger animals and artiodactyls all have ranges up in about the 0.75, even though it hasn't really been measured. It has not been measured in baleen whales and other cetacean species that are bigger than that can't be kept in captivity. So then we can substitute in our intake for our feeding rate times our engulfment volume, because we're assuming prey density is constant, and that's going to be proportional mass to the 0.75. And critically, those aspects, both engulfment volume and mass, can be allometrically to whale length. So if we relate those to whale length, which we can work out later on sometime, I'll show you the derivation of that, you can get an expected slope of your log-log plot of what you would expect that relationship of body length to feeding rate to be. And one of the things that you see is that this negative 1.59 here is much steeper than what we actually observed, the 1.18. So that what that suggests is that if we think that this is all, if we're happy with this derivation, that we think that minke whales are feeding less on average when they're actively feeding than we would expect from this derivation of lines, or you can think about it as blue whales feeding more than you'd expect, depending on your perspective. So how can we explain that? What's going on? Well, one of the things that we can look at is remember that the feeding rates vary quite dramatically during the day and at night. So if we break this down and look at actually the day feeding rates and the night feeding rates, you actually get a really good match between the night feeding rates and that expected line, and the day feeding rates are way lower off the charts, right? So what that starts to suggest to us is that the night feeding rates are kind of the critical component of these animals foraging, of minke whales specifically. Blue whales seem to be about the same for day versus night because they've got all these other adaptations to kind of account for that. Their large size kind of buffers them to allow them to dive deeper. So minke whales is this critical nighttime feeding component. And it's the same result, basically, if you think about that as a shallow feeding versus deep feeding, or day feeding versus night feeding, because at night, all that prey is shallow. Okay, so this is the big conclusion here. So minke whales, so this is just that blow up here of that minke whale foraging. So we have the day versus night. We have our expected lines there. The dashed line is the regression of what we'd expect to see. The solid lines are the regressions on the actual data. And we're going to start thinking about these temporal constraints. Right there is 4.5 meters is the length of a newly weaned minke whale. So that's about our minimum size at which an animal can feed by itself. And we have some animals tagged right in that size range, but that's about the minimum size we expect to see in the rorqual whale family there. Okay, remember we can break up our foraging rate into these temporal constraints. And we have this length of minke whales, 4.5 meters. Okay, so now we're going to look at the different components of lunge feeding on this time scale on the y-axis and body length on the left axis here, and look at how those different temporal constraints vary with body size. So firstly, we have the engulfment capacity, the time it takes to actually lunge and engulf that krill. And there is a relationship with body size. It increases with body size, but we're looking at the difference between 0.6 and 1 second. So it's not a big component. It's not a big part of that foraging cycle. It doesn't really matter that a smaller whale can lunge quickly, can engulf more quickly. Acceleration, how much time does it take for an animal to get up to that 4 meters per second, is a critical component. It takes about 5 seconds, but it doesn't really scale with body size either. It takes about the same amount of time for an animal to get up to that speed, to get up to about 4 meters per second. Filtration, as we've talked about, is a big component. Now we're talking about a difference between 6 and 10 seconds. It's a 50% difference between the small size and the large size, and you have a pretty strong relationship between body size and the amount of filtration time it takes. But then there's also this environmental component. This environmental component is how does this animal find food? And that search component is big for all of these animals, kind of regardless of body size. There's not really a strong relationship between how big you are and how you find your next meal. So then if you look at the overall amount of time it takes for every lunge, there's not that much difference between a small minke whale and a large minke whale, 26 to 30 seconds or so. And critically, the proportion of time that you spend on things that are manageable, like you can have a smaller filtration time by being smaller. That proportion of the overall lunge time due to things like filtration decreases as you get smaller. So remember, this is actual minke whales, but if we had thought about, oh, why isn't there a 1 meter or 2 meter minke whale? The amount of time that is proportionally that you can actually adjust by lunging more quickly or something, gets smaller and smaller and smaller. Okay, so what does that allow us to do? Well, it allows us to predict what the maximum feeding rates could be for any given animal. So if those two gray lines I just drew there are different depths that you're assuming that these animals feed at, and you can see that they kind of constrain the maximum feeding rates that you see for those nighttime feeding rates. Similarly for day, at different depths, you have a constraints on the maximum feeding rates for that daytime feeding animal. And if we extend this now to the left to below a 4.5 meter line, because of those different proportions of how those animals are spending their time, you get this curvature where you have this curvature starting to go away. Right here, you have a nice slope. At the big size, we actually have maximum feeding rates that match the observed slopes and the expected slopes of we'd see allometrically. And below 4.5 meters, you see a curvature, meaning that the current way that the rorqual whales are adjusted and the amount of time that they have to spend lunging at this food is not sustainable for animals less than 4.5 meters long. So they would not be able to get enough energy in the current ocean conditions to do this behavior. So that's kind of the point of what we're trying to get to with this, of why these animals, why aren't there animals smaller than a minke whale in today's oceans? Why aren't there rorqual whales doing this feeding modality? We're getting close to the end of time here. The follow-up questions would be, well, what's going on with blue whales? Why can't we have, why are blue whales also restricted? Why don't we have animals bigger than blue whales? And we'll just kind of hint at what we're working at here is this idea of physiological constraint. So this is a heart rate of a blue whale that we measured. And one of the critical findings here.
So thank you for following up.
Okay.
If you're online and you are unmuted, can you mute yourself? Thanks.
we'll wrap it up here in a second. But critically, the question is there a physiological limitation? One of the cool findings on blue whales was that when they dive, their heart rates dive down to 2 to 4 beats per minute. That's really cool. That's similar to other marine mammals. What the really cool finding was also that when they come back to the surface, their heart rates are near maximal. And we think that's happening because blue whale's heart actually takes nearly 2 seconds, about 1.8 seconds, to actually contract and beat. And so they can't beat much faster than 30 beats per minute, which they're doing on every dive almost regularly, about 30 beats per minute. So there's this idea that they can't recover fast enough, and they start to self-restrict themselves without some other adaptation to allow a faster heartbeat in bigger animals. And so that kind of gets us to this idea of, okay, well, like what could be going on here? Why looking at the right temporal scale allows us to start asking this question of what's the restriction here if that's so energetically efficient? Yeah. These last final questions that we're kind of looking at in the future, right, is can we parse when lunge feeding developed based on the fossil record? And can we use what we know now about minke whales and about this feeding modality to say something about primordial oceans? If these animals need to be 4.5 meters or bigger to make engulfment filtration feeding happen, when and how did that feeding mode actually devolve? And were the oceans slightly different, or was it a slow process and finally like, okay, we're both 5 meters big and evolving this engulfment filtration system? And then once you have this filtration system in place, that allows giganticism. But we're looking for kind of more record and more evidence of that as we go. And that leads to some ideas of niche separation, but we'll leave it at this for now and take any of your questions. So thanks for everyone's attention.
All right. That was amazing. Thank you very much.
Thanks, Adam.
So do we have any questions online? I'm going to start there. Not yet. Okay, for folks online, if you want to put any of your questions into the chat, we'll read those out, or go ahead and raise your digital hand. For those in the room, you are welcome to go to the mic or raise your hand and I'll bring you this mic.
Thanks for everyone being here. It's really just nice to see a full audience, and it's really fun to kind of start getting back into these communities. So it's neat to see everyone here. And
all right, well, I'll watch Minda walk to the mic to ask the question. Minda, that mic is not on yet. Will you turn it on? There's a little, like a slider. Now, is it on? Okay.
I was curious, you were talking about the different ways that the blue whales expel the water. Is there any, I never have seen any footage of, is that something that humans can see? How do we examine that?
Yeah, some of the best footage we have is from, so I think everyone heard the question, do you think, online and such? Yes, okay. So I won't repeat the questions, but some of the best footage we have is similar to what I showed you guys with that blue whale with the air coming out of it. And you can actually sometimes in various scenarios from divers and sometimes from our tag videos, you can see if the water is at all discolored, you can actually see those kind of plumes of water coming out the sides. And sometimes you'll see the lips kind of close and open as water sloshes back and forth. But that's our best evidence, and it's all snippets. And we don't have any like really full-scale studies with a big sample size to say exactly what's happening. But we do have just those small snippets of evidence.
All right, hang on, Ichang.
So on top, following that question, there was a diver that was swallowed by a whale on the East Coast. Has there been conversations with that person about what that was like? And okay, but kidding aside, the jaw structure, fascinating, great presentation, loved it. The jaw modification or the chin.
Yeah.
Is there any example of that in terrestrial organisms as far as a comparison or even just kind of a consideration of that coevolution?
Yeah, great question. As far as I know, that's a unique adaptation to rorqual whales. There is some other animals do have unfused mandibular synthesis, but as far as I know, I don't know any animals that have that similar kind of rotatable structure in that same way. There are things like pelicans that are kind of similar, where they can have that expandable, throat pouch, and when they dive down. I wouldn't necessarily say they're filter feeding, but they have that kind of same expandable pouch. And I don't think that they have that same mandibular synthesis idea. So, snakes, Yeah, snakes. I don't know about snake jaws. That'd be a good one to look into. I don't know if it rotates or if it just detaches or what. So that'd be cool.
I'm just going to pause real quick. Any questions online? Not yet. Okay, go ahead. Scott.
Okay, thanks, Dave. Great talk. Really interesting
Thanks, Scott.
ideas. I remember years ago hearing Rick Lambertson ask a question that we spend a lot of time on asking about the energy of opening the mouth and filtering in a baleen whales. But his question was, what about all the energy of keeping your mouth shut? How does a blue whale keep its mouth shut?
Yeah, great question. One of the things that we keep trying to narrow down the answer to is the degree to which a whale that is opening its mouth and engulfing all that water is actively resisting that inflow of water into it with its ventral groove blubber musculature. And it seems to be because there's enough drag, right, as you open its mouth just to inflate that VGB like a parachute. And it doesn't take very much drag. You can be going about 0.5 meters per second to actually just passively inflate that. But there is some resistance there based on the timing of how long this whole process takes. We think that they're actively using their muscles to kind of contract that whole process. The mouth itself has, there's basically huge muscles that connect to the back of the skull here. And I'm not enough of an anatomist just to tell you what muscles those are called, but there's huge muscles that connect back there that are, contract to help close that mouth and keep it shut. But that's all. That's the best answer I can give you.
All right. Any other questions in the room? 1 second. Hang on. We got a little line going on. That's great.
Yeah, thanks, Dave. That was very good. How does the length or the time duration of the individual feeding bouts compare to their estimated aerobic dive limits?
Yeah, great question. Aerobic dive limit is we go back and forth about whether that's to the degree to which we should try to figure out that concept. And it's most of the ADLs that I see tend to be estimates still in a lot of ways because it's hard to gauge that. And you probably know way more about that for gray whales than I do. And I think the thing that we're seeing most generally is that these animals are not reaching their aerobic dive limits and that they are below that limitation. I don't know. Do you have anything else? Do you have any ideas about that? We can talk later. Thanks.
All right, I'm going to hand the mic to Lisa.
Oh, I meant to say too. Yeah, what'd you say your talk next week is going to be on like the super rad component? Hopefully this is moderately rad. That's what I was going for.
Well, as a century ecologist, I've always been curious, but never really pursued it much. How in the world do they find the plankton? Like what sensory modalities do these animals use to find that? Especially at night. That's really the one I'm curious about. I mean, because they're super duper tiny and you're not going to be able to see it. And then, so anyways, just really curious.
Yeah, great question. And that is like one of the foundational questions that we kind of still get asked at almost every talk. And because it's a big question and we don't have a great answer for, there's kind of a couple of schools of thought, especially at night is a great point. Because for a while, like blue whales along the California coast and mostly humpback whales too, do most of their feeding during the day. We have some cool videos of them silhouetting, even at depth, they can silhouette some of the krill patches above them and kind of use that downweld light to help identify those krill patches. But then at night, what do they do on a dark night, moonless night? Like what do they do? And we have some evidence that they feed more often during full moons, but whether that's related to prey behavior or predator behavior, not sure yet. So how do they find it? There's kind of two schools of thought. One is some sensory modality. They might have good hearing. There is sound that comes from a deep scattering layer as these krill move up and down in the water column, and they have good low frequency hearing and can identify that. I kind of play around with this idea of acoustic daylight too. Have you thought about that idea? It's this idea where if you have enough of a obstacle, this comes from a script study where they just took like a hydrophone out to the pier and they just kind of moved it around, and you could identify where the pier is because it blocks the background noise. And so if you have a dense enough krill patch, you might be able to actually kind of identify that based on the gap in the background acoustic radiation. And then there's the third idea, which is just that they swim around until it hits them in the face, and then they start foraging there. And so they kind of mark that and know where to go back to. Yeah.
All right, one more question and then we'll wrap everything up.
Thanks, Dave. Fascinating talk. I love allometry. And my question is a quick one. Have you ever, or if you have not, have you ever considered adding a fourth species to your impressive data set? And if not, why not? And if so, could you give us a hint about how that fourth species falls in those plots?
Yeah, great question. So I had blue whales and humpback whales and minke whales, but there's some gaps in there. And there's a number of mid-sized rorquals that could help fill in some of those gaps. So do we have more species there? And that's certainly, let's write a grant, let's go do that. There's one of the answers. Those are the 3 study species we've had the most success with. Part of the other answer is fin whales would be a natural fit to kind of fit in there. Fin whales on the Atlantic side where we've done a lot of tagging. I could show you some videos, but those tags slip off really quickly. They have a lot of diatoms on their skin, tend to, and there's we call it the Stellwagen sleigh ride when it just kind of slides right off the back of the animal. So we don't have as much fin whale data as we probably should have, is one answer. And that would be a good thing to explore. I'd love to also do some Rudy's whale studies and sei whale studies, which have some different feeding modalities as well, and look at some of the different feeding ways to do that.
All right, I've been begged for one more question, but I want to make sure that there's no questions online. Okay.
Thanks, Dave. You know, when you imagine a blue whale at depth with his mouth open, he's got to get it closed again pretty quick. And, you know, it's this huge, long jaw bones filled with tons of water, and they kind of snap it shut pretty quickly. Is that all done with musculature at the rear of the jaw, or are they using some of the their position in the water to help, you know, some physics to help them close that?
Yeah, good question. I haven't done any modeling or to think about like the physics of the water column and how that works, because they tend to work. They do lunges in all different directions. You know, they'll sometimes do these 360 maneuvers. They'll sometimes lunge straight up, sometimes just on their side. And you know, it's all kind of neutrally buoyant. So I don't know that there is like a specific water orientation that they use. But in addition to musculature, there are you know, ligaments that kind of have some elastin properties as well that I don't want to go too much into detail because I don't know enough about it. But there are some kind of tensions that get pulled back together as well. So it's not just all muscles, but some forces like that too. But I don't know enough about that.
All right, I think what I'm going to do is if there are any other questions, we'll go ahead and end our presentation now, and you can go up and talk to our presenter, because I think we can linger for a little while. But I think we all heard it, right? Some opportunities for collaboration was thrown on the floor. So I'll just throw that out there. For everybody online, thank you so much for joining us, and please join us again next week. For everybody in the room, thanks for being here, and we will have an online and on-site option again next week. Yeah. So it feels good to have everybody back. I believe, unless the students have found them, there's still cookies as you head out the door. If you did not log in when you came in, can you please do that? That helps me justify buying our cookies. Just appreciate that. And then one more thank you. Thanks.
Oh, thanks, guys. Yeah. Bob, you should talk to Jeremy too. He's the one who can do all the anatomy. He'll answer your anatomy questions better than I can.
All right. For those
Thanks,
online,
everyone.
we'll see you next week. Thanks.